Hello, ladies and gentlemen, and welcome back to another Predictive Biotech presentation. My name is Arthur Hahn. I'm the Vice President of Marketing for Predictive Biotech, and we are joined here today by Beatrice Uscanga. Beatrice, how are you doing?
I'm doing well.
Okay, Beatrice. So what are we going to be talking about today? I know you've been dealing with a lot of other experiments in the house, and you've been doing a lot of really cool stuff, but this one, I have actually come to you particularly because I thought it was pretty exciting. So what is a wound healing assay?
So wound healing assay is an in vitro study, and we basically just measure the functionality of our products. And before I get into the what's and how's of the wound healing assay, let's talk about the difference between in vitro and in vivo.
Perfect. I always get this confused, and I know I bug everyone about it. So what are the differences between in vitro and in vivo?
So the biggest difference is that in vitro is not done with a human or an animal. So in vitro is done in a lab in a very controlled setting with test tubes, 6-well plates, flasks. In vivo is done in a human or an organism, or in any living organism.
Okay, perfect.
And before we move on, I just want to note that in vitro studies do not always represent the same thing that in vivo would. The results from in vivo would be completely different because there's always immune responses that are going to be different than what happens in vitro.
How would you even do an in vitro or in vivo study of a wound healing assay? Is there even one comparable?
Yes.
Just go and scratch somebody and put some product on them?
I mean, you definitely could do that, but I don't know the ethics behind that. There's actually a study that's been done on pig skin. So they've cut a box on a pig skin and then measured or tattooed the outside of the wound and then watched it close over time. And I mean, that's great, but we don't do any in vivo studies here at Predictive. So we stuck with in vitro.
Okay, awesome. So what is a wound healing activity assay? So, I mean, well, you explained it, I guess, earlier. It's just a scratch test. Is that what it is? So how do we start that in a lab? How do we even begin that?
Yeah. So like I said, it's in vitro. So we use a flask to grow our cells to confluence. And then once it's completely full of cells that are really healthy and ready to go, we don't want to overgrow them because then they will become too mature and they end up not working the same way. So we have to watch these very, very closely. It takes a lot of time, a lot of focus, a lot of checking. It's like having babies that you need to constantly check in the incubator.
So what do you mean by overgrow? Like, is there a certain age that they're good for this experiment? Do you have to pick out a certain period?
Yeah. So in actual skin, so we use keratinocytes in this study, by the way, and in skin, your keratinocytes actually move up through the layers of your skin. As they move up, they become less and less active. And by the time they get to the top layer of your skin, that's actually what sheds off.
So callus, basically.
Well, no, not necessarily callus, but the dead skin cells that come off that we all shed every single day. Those are far mature keratinocytes than we want to use in this assay. You want to use fairly young keratinocytes in this assay. So if you overgrow them, you're going to end up with a lot of dead ones that don't really have activity.
Okay, so how do we start this whole assay?
So the very first thing you do is pick cells. Pick what kind of cells you want to use and grow a monolayer. And then in this assay, the next step would be to scratch that monolayer. And we use a 200 microliter pipette tip to scratch each of the wells that have the monolayer.
That's really tiny.
Yeah, it's very small, but we use a 10x objective and a microscope to measure this. And I mean, if you've ever looked in a microscope at a 10x objective, you can see it fairly clearly. And it's in the same location every single time. And so what you do is just take pictures of that same spot and you watch it close over a couple days.
So is there a certain time of the day that you guys are actually taking these photos?
Yeah, so we actually took it every 4 hours, which was kind of tedious, but we hope the next time to run this with the live cell imaging so we don't have to set timers and we don't have to go back and keep taking these pictures because honestly, we're taking it in and out of the incubator and it's not great for the growth. So here's the control. So we use day 0, day 1, day 2, and these are all, you can see there's not a lot of wound gap closure. So this is just a typical wound. This would be all of the 4 phases involved in wound healing, which is hemostasis, inflammatory, proliferative, maturation. And since we're doing it in vitro, we're only able to use the proliferative phase and the reepithelialization cells.
So what do you mean by we're only looking at the proliferative phase? So we don't look at hemostasis, inflammatory. Is that not shown within these controls, or is that what you're considering in vivo as far as inflammatory response? That's what it is, right?
Yes. So hemostasis is technically when you have a clot. So if you don't even, so this is an epidermal, okay. This is an epidermal wound. So you wouldn't have any clotting at this anyway. So technically that wouldn't even be part of in vivo. However, proliferative would be, because that's when it basically, or inflammatory actually is the second phase. And that would be when you have a rush of cytokines to that area that allow all of the cells around and all of the tissue to swell. And it's usually to push out any of the foreign objects that are in there, any bacteria or anything, to prevent infection. And then the proliferative phase, it contracts the wound to try and pull back together all those fibers. And then there's the reepithelialization, which that's what we're testing here. And this is when all the cells start to fill in that wound gap closure. And
so it's a very specific time frame in the wound healing process that we're trying to measure in a laboratory.
Yes. Yep. And there's no other, you can't really have any other parts of the phases because there's no blood, there's no cytokines that cause the inflammatory response, and you can't really see any inflammatory response in these. I'm sure you could probably measure the secretion from inflammatory responses, but I don't know if that would be possible in this kind of assay. And so what we're measuring is the reepithelialization phase. And the way we do this is by adding our product polycyte, and we add that to the media as a 1-to-1 dilution, and we add it to each of the different wells. And as you can see, the wound gap closes much quicker than the control. This is likely because of a cell-to-cell interaction that's allowing them to fill the space quicker. So basically, they recognize the cells next to them and they say, oh no, we need to quickly fill up this spot and we need to grow and try and proliferate into this empty space. Based on our photos, as you can see, the control versus the wells with polycyte in them, our products have potential to increase that cell-to-cell interaction. And therefore, we can likely hypothesize that it is increasing in a speedy healing of a wound.
So when you're adding the polycyte, are you adding it at what time do you add the polycyte? Is it at the very, very beginning before do you do the scratch and then add the polycyte, or do you have the polycyte in there and then do the scratch?
So you do the scratch and then you rinse it with media with nothing in it. And then you add the media into it as a 1-to-1 with polycyte.
Okay. And the control has just media only.
Yes. Yep. The control is just 100% media. And at this point, we've only done full strength polycyte, but we would like to do dilution series of the polycyte to see if there's any change in how long this takes to heal.
So have you seen any difference between, the control is just media itself. Do they have is there other things that you can add to that initial control to make it match the speed as polycyte goes? I'm just kind of curious. Is there a growth, is there a miracle grow for cells they can put in there?
No, we've actually added several different growth supplements. There's tons offered by companies that you can add to your media to help with adherence, to help with cell-to-cell interaction, just different things like growth supplements. In this case, we have all the growth supplements in our media and in our control. And as you can see, it doesn't increase the speed of wound healing. And so the only thing that we have seen to work is adding our polycyte to it, and that increases it quite a bit.
And I heard you guys got a cool new machine to kind of bring it in, actually while we're going to make a real-time video.
Yeah, so it's actually a live cell imaging software that we have in our lab, and you need an onstage incubator. We didn't have this for this experiment, but we do have it now, and it's all set up and ready to go. And we've got a second round going that we're going to have live cell imaging for probably around the end of next month.
Awesome. So we'll have an actual, a cool real-time video of the wound gap actually closing with it.
Yeah. Mm-hmm.
I know you guys are running all different types of experiments, and especially with this wound healing assay. What is the next step?
We actually have a really cool piece of machinery here called MagPix, and it measures the amount of cytokines that we have in our products. And we actually use that as kind of our quantitative research. And this wound healing assay is more of our qualitative research. So it's like a second check. So we verify that we have the cytokines in there, and then we verify again with the functionality of those cytokines that were measured.
So after all that is done, what's the next level of experiment? Are you guys all done with the experiment now? Are you satisfied, or is there additional stuff that you're going to be looking for?
I feel as though in research, I'm sure anyone else could agree with this, you are constantly looking for answers. Even if you have the answers, you're still kind of trying to get the all-around, well-rounded picture of the answers. So there's always different angles you can look at it. There's always more data you can gather. There's always more experiments you can perform. It's just kind of like you want to triple check yourself. So anything that we do come up with, we like to check it again and again and again.
Well, Beatrice, that sounds awesome. I know you kind of gave us a quick whirlwind into your world here. But anyway, I just want to thank you for taking the time out of your busy day, and thank you all for listening. So if you have any questions, please visit our website at predictivebiotech.com or contact us via email, and all of our details will be in the description below. Hope everyone stays safe, and we look forward to you joining us very soon. And remember, better begins here.